All right, so the focus today is on these stages of cellular respiration. We're also going to spend a little time talking about what happens when you don't have access to oxygen. All right, so cellular respiration only happens in the presence of oxygen. We'll talk about what happens otherwise too. There are 3 major steps, but the second step has 2 parts. All right, so we're going to make all of our ATPs during these 3 steps. The first one is glycolysis, then pyruvate oxidation in the citric acid cycle, and then oxidative phosphorylation. So it's always in this order. There are a few really good figures in your textbook that kind of help organize where these things are taking place and how much energy they make. Right, so the first part, glycolysis, is going to take place in the cytoplasm, so outside of the mitochondria, and it is going to make a small amount of ATP. Then we're going to move from there into the mitochondria. So these 3, pyruvate oxidation, citric acid cycle, and oxidative phosphorylation, they all happen inside specific parts of the mitochondria. And you'll notice that most of our energy is going to be made in this very, very last step. So here we're going to be making the bulk of our ATP. So we're going to use ATP to power all of the other things that the cell is going to work on. The other thing we want to think about is these are coenzymes. So we talked about before how part of this process is going to use coenzymes. These work with enzymes to store electrons and hydrogen ions. And that is what we need to make for this phase, right? So part of what we're going to be thinking about is ATP. That's what we ultimately want to make. But to make all that ATP, we want to make coenzymes, NADH and FADH2. Okay, so this is a pretty good summary about where they are and how much energy they make. And most of that energy is going to come from here. So we're going to start talking about glycolysis. So glycolysis takes place in the cytoplasm. And that means that any cell that has cytoplasm is going to be able to do it. All right, so not all cells have mitochondria, right? Bacteria cells do not, but they do have cytoplasm. So all of the cells are going to do this step. And the step itself is long. So in your textbook, the image of the steps looks like this. It's a linear pathway. So remember, linear pathways start with an initial reactant, and they have a final product, and they have a lot of intermediates. Now, you are not responsible for everything in this chart. This is what I mean by your book has a lot of details that you don't need. So let's go over the parts that you are going to need. So the first thing we want to think about is, what is the initial reactant? And that is glucose, right? So in glycolysis, we are going to be breaking the sugar glucose. In order to do that, it is going to cost us some energy. So ATP is going to be used to convert glucose into other intermediates. So for example, if we want to get glucose all the way down here, it's going to cost us energy to turn it into glucose 6-phosphate. It's going to cost us energy to convert fructose 6-phosphate into fructose bisphosphate. So we need energy. You do not need to know the names of any of these intermediates, only that it requires energy to get there. Okay? So don't need to know those. That's going to get us down here to G3P. This is the only intermediate you need to know. And that's because it's going to be important for photosynthesis. So all the rest of these intermediate names, you don't need. They're gone. Just that one. The next steps are going to produce a few things. So we are going to make some coenzyme, and we are going to make some ATP. One of the things that is mentioned is that it is 2 net ATP. Does anyone know what the difference is between net and gross? Have you heard those terms before? Net and gross? Usually we think about them when it comes to money. No? So when you get paid at the end of the week, yes, gross is what you initially have before what? Taxes. See, you guys do know. All right, so net income is your take-home pay. It's what you actually get put into your bank account every week. That includes everything that gets taken out, i.e. the cost to your employer and to you for you getting paid. So when we think about the amount of ATP that we make, the gross value is 1, 2, 3, 4 ATP. That's the gross value. But it cost us 2 ATP at the start. That means that the net value, the actual increase in ATP that we take home, is going to be 4 gross ATP minus the 2 ATP that we had to pay. So we have a net gain of 2 ATP. Okay? So when we're saying that we produce 2 net ATP, we make 4 gross, but it cost us 2. So we're actually only increasing our ATP by 2. Okay, so that's the difference. At the end of the linear pathway, we are going to produce pyruvate. So that is our end product. So these are all the parts you need to know. So we can simplify this a lot, right? So we can simplify this a lot. There are a couple of ways to think about simplifying. So we know, for example, that glucose is our initial reactant and that pyruvate is going to be our end product. The other thing you should notice about this is we are essentially cutting glucose in half. This is the number of carbons that it has. So glucose is a 6-carbon molecule and pyruvates are 3-carbon molecules. So for every 1 glucose, we will make 2 of them. We're going to put in some ATP and we're going to get out 2. We're going to put in some NAD+ and we're going to get out some NADH. Another way to think about it is this. You could say glucose is up here. We're going to get to 2 G3P. This is going to cost us some energy. Then from there, we're going to produce pyruvate. We're going to get out ATP. And we're going to get out NADH. So instead of having that really big linear pathway, this whole thing, we can simplify that down to the parts we actually need, which is this. Whichever way it helps you to think about the pathway is fine. You can condense it as far as this one over here, or you can condense it here. So essentially NADH and ATP are end products, but we're not going to use them to move on to the next pathway. So we're gonna write all of the things that we've learned here onto our chart, and that'll make that clear, okay? Okay, so let's go put what we know onto the chart. So where does glycolysis take place? In the cytoplasm. And which one is our initial reactant? Glucose. And what are end products? They are 2-pyruvate, 2-ATP net, and 2-NADH. Does it cost us any energy to do this? Yes. Our net ATP is 2. And the only other note is G3P is an intermediate that you need to know. Not for this test, but for when we get to photosynthesis. Does that help make this really scary looking pathway not so bad? Yes. Okay, so that's the goal today. We're going to take all these big pathways and we're going to break them down to the details that you need to know. All right. Any questions about glycolysis before we move on? Okay. So glycolysis is the first step and it's universal. All cells do it. The remaining steps, pyruvate oxidation, the citric acid cycle, and oxidative phosphorylation, are going to happen in the mitochondria. And they happen in different parts of the mitochondria. So this means that organisms that don't have mitochondria, like bacteria, cannot do these steps. So now we're going to move into the mitochondria and we're going to talk about all 3 steps and what happens during them. Right? So I'll remind you that 2 of these steps are very closely linked. So pyruvate oxidation and the citric acid cycle are very closely linked. And then a lot of the things we make during these first 3 cycles are going to then be taken into oxidative phosphorylation. So in this image that you're looking at, what it's telling you is that the glucose got converted into pyruvate. That pyruvate is then going to get converted into acetyl coenzyme A, and that is going to go into the last pathway. Lily, get off my computer. Thank you. From there, the only thing that will connect us to the oxidative phosphorylation pathway is the coenzymes, right? So we're going to do these guys first. So this is the fate of glucose. The first step in this process is the pyruvate oxidation cycle, well, step essentially. Both of these, pyruvate oxidation and the citric acid cycle, are going to take place inside the mitochondrial's inner compartment. So remember, the mitochondria is made up of two membranes. So here's the outer membrane, and here's the inner membrane. There are two compartments. This is the outer compartment. And these steps are going to take place in the inner compartment. The first step, pyruvate oxidation, is one step. We are going to take every pyruvate and we're going to convert it into acetyl coenzyme A. Notice that we are going from a 3-carbon molecule to a 2-carbon molecule. The carbon is lost as carbon dioxide. So the main things we're going to make here, we're going to make NADH, we're going to make acetyl coenzyme A, and we're going to make carbon dioxide. Our initial reactant is going to be pyruvate. Do you see any ATP in here? No. So we're not going to make any ATP, and it's not going to cost us any energy. Right, so we can add that to our chart. So where are we going to do this? Where in the mitochondria? The inner compartment. Of the mitochondria. What is our initial reactant? Pyruvate. And we're going to make all right, we're going to make one NADH. It's step 2. We're going to make some carbon dioxide, and we're going to make acetyl coenzyme A. There's no ATP involved, so it doesn't cost us anything, and we don't make anything. And there are no notes. Nothing fancy happens here. Now from here, the carbon dioxide will go to your lungs and it will go out when you breathe. So this is one of the sources of carbon dioxide in your body. We're going to save the NADH for later, and the acetyl coenzyme A is going to go into the next step. Right. So one of the things I'd like for you to notice while we're filling this out is here was our product. Now it's become the reactant. The next step is a cycle. So we've talked about this idea that linear pathways have an initial reactant, they have a final product, they often have intermediates, and they don't have to regenerate anything. But cyclic metabolic pathways do. So the citric acid cycle is not a linear pathway. It's a cyclic pathway. This changes the way we think about what is happening and what we need to have. So this is our cyclic pathway. That means we have two initial reactants this time. We have the acetyl coenzyme A that we got from the pyruvate oxidation cycle. And we also have oxaloacetate. So there are two things we need to make this work. From there, we're going to produce citrate. This is where the citric acid cycle gets its name. This is citric acid. You do not need to know the name of any of these intermediates. Only citrate. We are going to get out a lot of things. So we're going to make, here's one NADH, here's two, here's three. We're also going to make some carbon dioxide. We're going to make an FADH2. And do we make or use any ATP this time? Yes, we do. So we're going to make an ATP down here. What is our end product this time? So we're going to make all these things, but what is our ultimate end product? So remember, this is a cyclic pathway. That means that one of our initial reactants is also our ultimate end product. If we don't reproduce oxaloacetate, this cannot continue. So we are going to make some ATP in the process of going through all of this. But our ultimate end product is oxaloacetate. If we don't remake that, we cannot keep doing the cycle. We won't be able to make any more ATP. All right, so we're going to review that one more time, and then we're going to put it on our chart. We're going to take our pyruvate and move it into the inner mitochondrial compartment. Coenzyme A is going to come in. We'll make acetyl coenzyme A. That's pyruvate oxidation. And we're going to get out some NADH and some carbon dioxide. We're going to take our acetyl coenzyme A and put it into the citric acid cycle. From there, we're going to produce some CO2, some ATP, and some NADH and FADH2. We also are not balancing this. Okay, so we actually make a lot more NADH2 than that, NADH than that. Now, on the chart I showed you before, it said that this step makes 2 ATP. We're making 1 ATP per pyruvate. per acetyl coenzyme A. But how many pyruvates do we have for this process from a single molecule of glucose? How many pyruvates did we originally make? 2. So for every glucose, you are going to go through this process twice. Once for each pyruvate. So you're going to make a total of 2 ATP, even though each pyruvate only gives you 1. So we're going to put it on our chart. So where does the citric acid cycle take place? Same place or new place? Same place. Ooh, turn back to black. The inner compartment of the mitochondria. We have 2 initial reactants this time. What are they? So CoA is in here. Yeah, acetyl CoA. Right? So we're going to take our acetyl CoA that we made last time. We're going to put it in here. And oxalacetate. What are all the things we're going to make? And you don't need to know the correct number. Just what are the types of things we're going to make? All right, we're going to make some CO2. We're going to make some NADH. We're going to make a little ATP. So citrate is an intermediate. We are going to make that. But I want the end products. We're going to make some FADH2. Yep. And one last thing. That's right. We're going to remake oxalacetate. Does this cost us any energy to make all these things? Did we see any ATP going in? No, we did not. We didn't put any ATP in, but we did make some. So it's 1 per pyruvate that was used. So 1 per acetyl coenzyme A, 2 total. So we make lots of stuff this time, and we make a little bit of ATP. Now we're not going to take oxalacetate and use it in oxidative phosphorylation. It's just going to go back into the citric acid cycle. So this chain of using our products as our initial reactants is ending. Instead, what's going to happen is we're going to take all of these and these guys are going to go over here. We're going to be using them for oxidative phosphorylation, right? So Normally, when we're thinking about cellular respiration, our focus is down here on ATP. But the goal of these steps is not actually to make ATP, it's to make these coenzymes, and we're going to use those coenzymes to make a lot of ATP. So that's what we're going to do next. We're done with these 2 cycles. We're going to take all that stuff over to our oxidative phosphorylation. Now, this also takes place in the mitochondria, but not in the same location. Not in the same location. And while you're studying or working over things at home, there is a video in the slide deck about what this looks like. But I'm going to walk you through drawing it, what it looks like. All right, so we're going to talk about it first, and then we're going to draw all the steps in order. And then we're going to put it on our chart. So this is going to take place inside the mitochondria. We're going to be using the coenzymes to carry electrons and hydrogen ions to our electron transport chain. Instead of happening in the inner compartment, this happens on the inner membrane. So this is going to happen on the inner membrane. And it's going to look something like this. And this looks really complicated, but it's not so bad. We're going to break it up into all the steps. In an electron transport chain, the goal is to move electrons. So that is what we are going to be focusing on. So the goal is to move electrons. Every time you move an electron, that is going to cause a release of energy. And we are going to use that energy to actively transport hydrogen ions into the outer compartment. The goal of this process is to set up a hydrogen ion gradient that is high outside and low inside. So the whole purpose is to set up this gradient. We are going to get our electrons and our hydrogen ions from breaking NADH and FADH2. We're going to drive the movement by using oxygen to accept the electron at the end. If oxygen isn't present, then the electron transport chain will end. So oxygen and electrons at the end of the chain will also bind to hydrogen ions, making water. This is the reason you need to breathe. The primary use of oxygen in your body is for this electron transport chain. When we make water, we're also reducing the number of hydrogen ions in the lower compartment. So not only are we moving hydrogen ions out of the lower compartment, we're also removing them by making water. So both of those are going to help us. The hydrogen ions want to go back to the area of low concentration. Remember, molecules always want to move from areas of high concentration to areas of low concentration. Why can't they automatically move across the membrane? What is it about them that prevents them from moving by themselves across the membrane? So the phospholipids make up the membrane, and we talked about types of transport last week. So molecules can move through the membrane by diffusion. But there's something about the hydrogens that means they can't do that. So what kinds of things can move through a membrane by diffusion? That's correct. So small molecules can move through the membrane, and hydrogen is a small molecule, but this is a hydrogen ion. It has a charge. Only uncharged molecules can move through the membrane. So the hydrogens are stuck outside. What we're going to do is we will use facilitated diffusion to harness their movement. So they want to move back. They want to move from high to low. They cannot move by themselves. So we have a special protein called ATP synthase that allows them to move across the membrane by facilitated diffusion. It also has an enzyme attached that is activated by kinetic energy. As the hydrogen ions move, that energy is used to make ATP. So every time the enzyme gets energy from movement of a hydrogen ion, it's able to make ATP. And that is how we make so much glucose. Right, so that is a summary of what happens. Now we're going to do it one more time, drawing all the steps as we go. And then we're going to add it to our stuff. So we're going to draw all the steps as we go. There are a few things you need for an electron transport chain. The first thing you need is you need a membrane. The reason you need a membrane is that it is selectively permeable, and the goal is to create an ion gradient. Ions cannot move through the membrane without assistance. To help them move, you're going to need proteins embedded in the membrane. One of the main functions for membrane proteins is to help things be transported through the membrane. When you want to transport things from low concentration to high concentration, that costs you energy. That's active transport. We want to actively transport hydrogen ions from inside the compartment outside the compartment. To do that, we are going to take our coenzymes, NADH and FADH2. These are carriers. Remember, coenzymes carry electrons and hydrogen ions. That's their job. The electron is going to go on a trip. When the electron creates a small bond and then breaks it with this transport protein, that is going to pump a hydrogen ion outside the compartment. As the electron continues its trip, every time it interacts with one of these proteins, that will provide enough energy to transport another hydrogen ion out of the compartment. At the end of the trip, it needs somewhere to go. Here, oxygen is going to be waiting to pick up the electron. And oxygen and hydrogen ions plus that electron are going to form water. Now that we've created a high concentration of hydrogen ions outside, those hydrogen ions want to go back to the low end. The movement from high to low is the way molecules want to move. So they will move back through ATP synthase by facilitated diffusion. So this part over here, this is active transport. And this movement over here is going to be facilitated diffusion. Every time a hydrogen molecule moves or ion moves, that is kinetic energy. It's physical movement. And that energy is going to be harnessed to convert ADP and its phosphate into ATP. When you're thinking about how this works, it's very similar to thinking about an air turbine. In an air turbine, wind is going to push on the turbine, causing the blades to spin. That's then harnessed to produce electricity. So we're converting kinetic energy here into electrical. We're converting kinetic energy here into chemical. So you can think of it like that. The movement of the hydrogen ion is pushing the enzyme. And every time the enzyme spins, that allows it to create an ATP as it converts the kinetic energy of the movement of the hydrogen into chemical energy, the bond between ADP and the phosphate. Now for this one, it's not as straightforward. So when we're thinking about how to turn this into our chart, we need to make a few changes. I want to make a few changes. So instead of thinking about what our initial reactants are, we want to think about who is the electron donor. So where does the electron come from? And the electrons are going to come from NADH and FADH2. We also want to think about who is the electron acceptor. The electron acceptor is who waits at the end of the electron transport chain to pick up the electron as it's moving. If they aren't there, then the electron isn't going to leave home and go on its trip. So the electron acceptor is oxygen. We have several different products that we make. So we're going to make water. We're also going to make NAD+, FADH+, and ATP. For this exam, you don't need to be able to talk about this all that much, except for how it connects to fermentation. We'll talk about that next. This process does not cost us ATP. We're getting all the energy we want from the electron donors up here, so it doesn't cost us any ATP. And it is going to make us 28 ATP per molecule of glucose. For notes, it's a really good idea to know the types of transport used. So remember, we're using active transport to move the hydrogen ions out, and we're using facilitated diffusion to allow them to come back in. Now, this is a lot of information. Are there any questions about any of these steps at this time?
Can you go over I think it's the last two steps, the products one more time?
Sure. So for oxidative phosphorylation, we're going to make 4 things. We're going to make water. So that's going to happen when oxygen catches the electron at the end. So that's this step right here. We're going to end up with NAD+ and FADH+ because when we remove their electron, that is what we're left with. Now, those are going to go right back into all the other reactions so that we can make them again. Remember, these are coenzymes. They're not really being changed all that much by this process. They're just losing an electron. The primary thing we're going to make is ATP. All right, so when you're thinking about things that we make here, what I want you to think about is you're going to make some water because that is what you get from accepting the electron. And you're going to make some ATP because that is the end goal. You do technically make these other 2 things, but they aren't end products. They're going to be important when we talk about fermentation in a few minutes. So I want you to know that you do make them here, but only because we're going to need them for fermentation. Okay? And I'll explain exactly what you need to be able to say about them in a couple minutes. Is there anything else you'd like for me to clarify before we talk about what happens when you don't have oxygen? For this last step where all this ATP comes from. Do you feel like you know what I expect you to know and what you don't have to know? And roughly how all the steps work? Okay, I try to be very clear because the book has a lot more detail than you need to know. Does everybody have a copy of this chart that we made together? This will really help you when you're trying to study for this topic.